Okay, so we're going to do the lecture questions for energy, enzymes, and metabolism. So the first one I want to go through is just, can you guys give me some examples of both kinetic energy and potential energy? So give me, what were the 3 examples of kinetic energy that are important in biology? So give me 3 examples of that in biology. Yep. What's that? That's true, but I guess I was thinking more of like inside the cell. You said, what'd you say? Yeah, heat's one. Light. And mechanical. So the mechanical one would be the moving, the physically moving something. And then what were 2 examples of potential energy that I gave you guys in terms of being important in biology? Right, concentration gradient and chemical energy, right. Okay, so chemical energy and concentration gradient. And then connected to that, electrochemical gradients and electrical gradients are forms of potential energy. So then we also talked about 2 different types of reactions. We talked about energonic reactions and exergonic reactions. And I'm going to break it down for you, but I want to know some differences between the 2. So one thing we talked about is the fact that you can categorize an energonic reaction so you can categorize a reaction as one or the other based on its change in free energy or its delta G. So which one has a delta G of less than 0? No. Exergonic. So an exergonic reaction is going to have a delta, so exergonic is going to have a delta G of less than 0, and then an endergonic reaction is going to have a delta G of greater than 0. Another difference is whether the reaction requires the input of energy or it releases energy. So which one requires the input of energy? Right, endergonic. And then exergonic, maybe you can just remember this, something is exiting. So in an exergonic reaction, you're releasing energy. And then the last difference that you should know is whether it happens spontaneous or it's not spontaneous. So which type of reaction occurs spontaneously? Right, exergonic. So an exergonic reaction will happen spontaneously. Endergonic reactions do not happen spontaneously. So can someone, for the next question, describe to me what a coupled reaction is, or what happens in a coupled reaction? So energy is released, but what happens in a coupled reaction with the released energy? Right. So in a coupled reaction, it's a pair of reactions where one reaction releases energy that drives the other reaction. And so when you're talking about coupling reactions, you couple an exergonic reaction to an endergonic reaction. The exergonic reaction releases energy, and that drives or provides the input of energy that the endergonic reaction requires. The last question is, that's a part of number 4, is what specific exergonic reaction did we talk about in cells that is often coupled to endergonic reactions? Right, the hydrolysis of ATP. So, and the hydrolysis of ATP, again, is the exergonic reaction, and that exergonic reaction is coupled to many different endergonic reactions inside the cell. Okay, so number 7, what occurs in a redox reaction? So I don't think I explicitly said it out loud, it was on the slide, but a redox reaction is the short way of saying an oxidation-reduction reaction. So what occurs in that type of reaction?
You said it's the oxidation reduction.
Yeah.
An oxidant loses an electron in reaction, or loses an electron and something gains?
Right, so you get the transfer of electrons. So you either get a partial transfer or a complete transfer. So one reactant loses electrons and the other reactant gains electrons. Okay. Number 18, what were the two environmental factors that we talked about that can affect enzyme function?
pH and temperature.
Right, pH and temperature. And what might changes in pH and temperature do in order to affect enzyme function?
Denature.
Right, denature. Right. Okay. So 24, A and B are depicting two different types of inhibitors. What type of inhibitor is being depicted in box A? Right, competitive inhibitor. And then in box B, that's a non-competitive inhibitor. So number 25, which of the following correctly represents the steps in an enzyme catalyzed reaction? Yeah, D. So you have enzyme binding with the substrate to form the enzyme substrate complex. Substrate is converted into product, and then the enzyme releases the product. What about 26? An enzyme speeds up a chemical reaction in the cell, but it can only be used once. That's false. So they can be used over and over again. They can catalyze the same reaction over and over again. Okay, 28. Which of the following is true for a reaction that has a delta G of less than 0? Right, E. So B and C. So yield means to produce, so the reaction will release energy, and then it's also spontaneous. What about 29? Which is true of the following reaction? So taking ADP, adding a phosphate group to make ATP. Right, B. So it has a delta G of greater than 0. If it was asking about the reverse reaction, where you hydrolyze ATP, then you would answer A, C, and D. So the hydrolysis of ATP is exergonic, so it has a delta G of less than 0. We already said that that can be used to drive endergonic reactions, and that also releases energy. Okay, so number 30 is asking about enzymes. Which of the following statements regarding enzymes is true? Yeah, B, that enzymes increase the rate of a reaction. What's that?
No, you look at them, all of them, like almost correct.
Oh, yeah, so I'll go through them. So what enzymes don't decrease the amount of free energy change. So the free energy change is the difference between free energy and the free energy of the reactants and the free energy of the products. And enzymes don't change that energy. What do they change? Right, the activation. And what do they do to the activation energy? Right, they lower that. So they lower the activation energy, but they don't actually change the free energy change that occurs during the reaction. Enzymes are not going to change the direction of a chemical reaction. We didn't say it. This one you should have been able to get right because you should know that B is true. Enzymes, you should know D, that enzymes they are not permanently altered by the reaction they catalyze. So I told you that they remain unchanged by the reaction. And enzymes do not prevent changes in substrate concentrations. Okay, so then 31, for the generalized equation where E represents an electron, which of the following statements is true?
E.
Right, E. So molecule A is oxidized because it loses an electron. So remember, oxidation is loss. And then reduction is gain. So B is reduced because it gains an electron in that reaction. Okay, this one I didn't explicitly tell you, but you just have to think about it. So in the biochemical regulation of metabolic pathways, how would one overcome the effects of a competitive inhibitor on enzyme activity? Right, increase the amount of substrate. So if the two are competing for the same site, if you add a higher concentration of substrate, then you're going to increase the likelihood that it's the substrate that binds to the active site and not the inhibitor. So one way of overcoming a competitive inhibitor is just by increasing the concentration of substrate so that it's more likely the substrate will bind than the inhibitor.
What are some examples of, you said there was some that were like poisonous?
Oh, I don't have any specific examples for you. Sorry. The one that I thought was sarin gas, that's poisonous, but I can't remember. It does inhibit an enzyme, but I can't remember if it does it competitively or non-competitively. Okay, do you guys have other questions within this topic that you guys want to go through?
What's the Vmax again?
So the Vmax is the maximal velocity of the enzyme catalyzed reaction at saturating substrate concentrations. So against the maximal velocity of an enzyme catalyzed reaction at saturating substrate concentrations. Do you have other questions that you want to go through from this topic? Okay. So the next topic that we're going to talk about is called cell communication. You can also call it cell signaling. So another name for cell communication is cell signaling. And what we're talking about in this topic is how does a cell respond to its environment, and then so how does it respond to signals in its environment, so how does it detect them, how does it interpret them, and then how does it respond to signals. And we'll see that in some cases with the cell can be responding to changes in things in terms of its nutritional environment, and then it's responding to that. In the case of multicellular organisms, one thing that's going to be important is the ability of two cells to communicate with each other. So what we're going to see in cell signaling is that one cell can release a signal that communicates to another cell in the body. Okay, so in terms of why is it important, so one reason that cell signaling is important is because it allows a cell to respond to its environment. So cell signaling is going to allow a cell to respond to its environment. So this is an example of that. So this is showing you a yeast cell, and what they're showing you here is that what changes in the environment is this yeast cell has no glucose in the environment, and then all of a sudden you have glucose present in the environment. And what happens in this case is some of the glucose binds to receptors. So the receptors recognize the glucose, and that causes the cell to put in its membrane transporters that will allow glucose to enter the cell. So it detects it through this receptor, and then it's going to relay the signal to the inside of the cell. The cell puts these transporters in its membrane, and then it can take up the glucose in its environment, and it uses it as an energy source. So in this case, it's essential for the cell to respond to a change in the environment. And then it's also important for cells that need to communicate with each other. So that's what they're showing you in this case. So they tell you that cells in the growing shoot tip sense light. They send a signal that's called auxin, you don't need to remember that, to the non-illuminated side of the shoot. And then cells located below the growing tip receive the signal and elongate, thereby causing a bend in the shoot, and this way the tip grows toward the light. So that's an example of what's called phototropism in plants, and that involves the communication of two cells within the plant. Also in your bodies, when neurons communicate with different types of cells within your body, that's going to be done through the neurons releasing a signal, and then the ability of the cells that they're communicating with to respond to that signal. So one specific example will be there are your somatic motor neurons in your body communicate with your skeletal muscle cells, and cause them to contract. So when they release the signal, the muscle cells respond by contracting in response to that signal. So the two things that are important about cell communication is it allows a cell to respond to changes in its environment, and then it also allows two cells to communicate with each other. Okay. So a lot of cell signaling pathways, not all, are going to involve the following components. So not all, but many cell signaling pathways will involve these components. So it's going to involve something called a receptor. And the receptor is going to be the part of the signaling pathway that recognizes the signal. So whatever the signal is, the receptor recognizes that. And then we're going to see that when the signal binds to the receptor, that activates that receptor. And then you have what's called a signal transduction pathway. So once you activate the receptor on the inside of the cell, you'll have a signal transduction pathway. And this consists of a series of components that are going to relay the signal from the activated receptor to what are called effector proteins. Okay? And those are the proteins that are going to carry out the change that happens inside the cell. Okay, so you have a receptor, a signal transduction pathway, so this transduces the signal from the receptor to the effector proteins. And then the last part is a change in activity of the effector proteins, and then it gives you some examples. It could be that the effector protein is a metabolic enzyme that's either turned off or on by the activation of the receptor. So you get the receptor binding, the signal gets relayed, and then this enzyme gets turned on, and then that enzyme now is performing a function it wasn't before. It's performing a function that it did not before it was activated. It can also affect genes that regulate proteins that regulate genes. These would be specifically called transcription factors. So a signaling pathway may end up causing different types of proteins to be made in the cell. And then it can also affect cytoskeletal proteins, and that can affect the structures within a cell, or whether the cell is going to contract or not. But you have those three components: a receptor, a signal transduction pathway, and then effector proteins. Okay. So let's talk about the first component briefly, and we'll come back to talk about different kinds. So we said that the way that a cell is going to respond to whatever the signal is through a receptor. And most receptors, as it says in the slide there, are transmembrane proteins on the cell surface. But there are also examples of receptors that are on the inside of the cell. Examples in your body are steroid hormone receptors. So an example of that would be estrogen. Its receptor is on the inside of the cell. Okay, so most receptors are in the membrane where they bind the signal, but there are also some receptors that are on the inside of the cell. So in that case, the signal needs to go into the cell and bind to the receptor on the inside of the cell. Okay, there's also different kinds of cell signaling. And they're all on here. So you have contact dependent, which is up in the left-hand corner. You have paracrine signaling. Then you have something called synaptic signaling. And then you have something called endocrine signaling. So we'll start with contact dependent signaling. So what happens in this case is that you have a signaling molecule that's bound to the surface of one cell that binds to a receptor that is bound to the surface of the cell that it's communicating with. So in this case, the signaling between the two cells is going to require that these two cells contact each other in that way. So the signaling molecule is tethered to this membrane, and then the receptor is tethered to this membrane, and then that's what happens in contact contact-dependent signaling. Paracrine signaling is when one cell releases a signaling molecule that goes and binds a receptor on a neighboring cell. Okay, so paracrine is when a cell releases a signaling molecule and then that signaling molecule acts on a neighboring cell. So this would be a local signal. One cell releases, the other cell responds to it. Synaptic signaling is actually a specialized form of paracrine signaling. So synaptic signaling is a specialized form of paracrine signaling, and it is how neurons communicate with other cells. So synaptic signaling is specifically used by neurons, and it's how they communicate. And in this case, what happens is a portion of this cell comes in really close contact with the cell that it's communicating with, and this portion of the neuron releases the signal, and then it binds to this neighboring cell and activates that cell. Okay, so synaptic is just a specific form of paracrine. And then the last form is endocrine, and endocrine is a form of long-distance signaling. So endocrine is a way of signaling over long distances in a multicellular organism. And the signaling molecules that are used in endocrine signaling are specifically called hormones. And in animals, including us, the way that endocrine signaling works is a cell will release the hormone, which is the signal. The signal enters the blood, and then the blood carries it throughout your body, and then it'll leave the blood and bind to a target cell at a distant site in the body, and then that target cell will respond to that signal. So in this case, this long-distance signaling in animals involves the blood because the molecule is released from one cell, enters the blood, travels through the blood to its target cell. Once it reaches its target cell, it acts on that target cell. Okay. Just to go back to this slide and say something that I didn't explicitly state, but in order for a cell to respond to a signal, it has to be expressing the receptor for that signal. So the example that I just gave you is that in endocrine, a hormone is released into your bloodstream. It goes everywhere in your body, but it's only going to act on cells that express the receptor for that hormone. So a signaling molecule is not necessarily going to affect every cell in the body. It's only going to affect a cell that expresses the receptor for that signal. So in your bodies, one reason that one cell may respond and not another is because the cell that doesn't respond doesn't express the receptor, so it can't recognize that that signal's been sent. Okay. So another thing that we mentioned in terms of cell signaling pathways is that activation of the receptor is ultimately going to lead to some response by the cell. And there's certain responses that are very fast or rapid, and there's other responses that are slow. And just in general terms, a fast response usually results in just altering a protein's function. So the protein is already in the cell, and the activation of this pathway just results in altering the function of that protein, and that's going to be a rapid response because the protein is already there and you're just regulating its activity. A slow response would be if you actually get the change in gene expression. So you get the activated receptor, then you have to make the RNA molecule, then you have to make the protein that the RNA molecule codes for, and then the protein, the new protein you make, carries out the response of the cell. So that's a slower response where you actually have to make the new protein, and then the protein can function inside the cell to carry out the response in the cell. So, and both can happen with the binding of one signaling molecule to the receptor. So in some cases you get the activation of the receptor and you get both rapid responses and slow responses to that activation. Okay, the next thing that I just want to cover is something we've already discussed, and that's how gap junctions allow neighboring molecules to signal to each other. So in this case, the signaling molecule can diffuse from one cell into the other cell through the gap junction, and then that's the way that a cell can directly communicate with another cell. Okay, another important thing, these are kind of just important things about signaling pathways that we're going through first, is that a cell isn't necessarily only responding to one signal. In a lot of cases, it's responding to multiple signals. And then what it's going to do is the response of the cell depends on what are the different signals that it's receiving. So what they're trying to illustrate by the slide is that when this receives signals A, B, and C at the same time, then that cell just simply survives. It stays alive. But if it receives A, B, C, D, and E, it grows and divides. But it would take all of these acting at the same time. So a cell is going to change how it responds depending on the signaling molecules that it's receiving from its environment. So if you change the different types of signaling molecules or change the signals that the cell is receiving, that's going to alter the cell's response to those signals. Okay, another important thing to realize about cell signaling, and this refers to multicellular organisms, is that different cell types may respond to the same signaling molecule in different ways. Okay, so you have the same signal, but different cell types are going to respond to that signal in different ways. And there's multiple reasons that this could happen. So for some signaling molecules, there's actually more than one type of receptor that cells can express for it. So their response may depend on the type of receptor that they're expressing. One example for that is acetylcholine that they're showing here. So acetylcholine, when it acts on your heart muscle cells, it causes a decrease rate and force of contraction, whereas on skeletal muscles it causes an increase in the rate of contraction. And the difference in this case, one of the reasons it's different is the receptor that the signal binds to is different between those two cells. Another reason that a cell can respond to this, different cell types can respond to the same signals in different ways, is because the signal transduction pathway that gets activated can be different on the inside of the cell. So on the inside of the cell, the signal transduction pathway that relays the signal from the receptor can be different depending on the cell type that you're talking about. And then also a third reason that it could be different is that the effector proteins that their activity is changed, that then alters the cell's response, can be different depending on the cell type that you're talking about. So, so versus heart muscle may be expressing one type of activator, sorry, of what do I call them? Effector protein, sorry. So this receptor may activate one type of effector protein in this cell, but in here it affects a different type of effector protein. Because you can see that this results in contraction, but here the signal binding to this molecule results in the release of another signaling molecule from that cell. Those two different responses would involve different effector proteins because the contraction involves the cytoskeleton, whereas release is going to involve making these vesicles fuse with the plasma membrane. Okay, so those are three different, different cell types can respond to different cell types can respond differently to the same signal, and I just gave you three reasons why that can happen. Okay, also another thing to realize about cell signaling in multicellular organisms is the actual concentration of the signaling molecule can affect how the cell responds. And so when they say morphogen gradient, what they're trying to, the yellow arrow is supposed to represent the concentration of the signaling molecule, so it's highly concentrated here. And then as you get away from the source of the signaling molecule, the concentration of that signaling molecule is going to go down. And these three different cell types are going to respond differently because they're receiving a different concentration of the signaling molecule. So the concentration of the signaling molecule also matters in terms of how a cell is going to respond to that, to the signal. Okay, so then now let's move on to talking about how cells actually respond to signals. And this is just kind of spelling out something that we've already talked about. So we said that one thing that you need for a cell to respond to a signal is a receptor. So the first thing that's going to happen in terms of a signaling pathway is that you get receptor activation. So what's going to happen in this case is the signaling molecule binds to the receptor, and then that activates the receptor's function. Okay, so signaling molecule binds the receptor, that activates the receptor. Then after you have receptor activation, you have signal transduction. So that's the signal transduction pathway that we're talking about. And what the signal transduction pathway does, it consists of several different proteins, and they're responsible for mediating the transduction of the signal or movement of the signal from the receptor to the proteins that are actually going to be involved in the cellular response. Okay, so you get the receptor activated. The receptor activation first turns this molecule on, which then turns this molecule on, which then turns this molecule on, and then this molecule communicates with the effector proteins. Okay, so you have receptor activation, then transducing or moving the signal from the receptor to the effector proteins, and then the third part of a signaling pathway would be the cellular response. Okay, so those are just three different parts of a signaling pathway: receptor activation, signal transduction, and then cellular response. And then again, it's just giving you on the slide there different ways that a cell may respond to the activated receptor. Okay, do you guys have any questions just about, in general, what's going to happen during a signaling pathway?
What was the definition you gave for signal transduction pathway?
Yeah, so a signal transduction pathway is a series of components. So a signal transduction pathway consists of a series of components that relay the signal from the receptor to the effector proteins. So it's a series of components that relays the signal from the receptor to the proteins that are actually involved in the cellular response. Which
After that, the proteins essentially react according to the signal.
Right. Okay, so we're going to take each of the different parts and talk about them in a little bit more detail. So we're going to start with the receptor part. So, and we're first going to talk about just some vocabulary that's useful to know with respect to receptors, and then we're going to talk about three different types of receptors. And we're going to focus on cell surface receptors, so we're not going to focus on receptors that are found inside the cell, so we're focusing on cell surface receptors. So the cell signaling molecule that binds to a receptor, or said differently, the cell signaling molecule that the receptor recognizes is called a ligand. So Ligand. That's simply the molecule that binds to the receptor to activate it. So you might recognize that word from ligand-gated ion channel or ligand-gated channel. And a ligand-gated channel acts it opens up or responds when the signaling molecule binds to it. Okay, so and what happens in terms of this interaction is this binds non-covalently to the receptor. So when it binds there, that's not permanent. It will on-bind. So it binds to it, that leads to the activation of the receptor. But once this molecule on-binds, it reverts back to its inactive form. Okay, so again, receptor activation is dependent on the ligand binding to the receptor. Okay, so what we're going to do is talk about 3 different types of receptors. The first type is going to be called enzyme-linked receptors, and that's what's being, so the ones that you're looking at on this slide are enzyme-linked receptors. Okay, and enzyme-linked receptors are going to typically have 2 important parts. So one part of the receptor recognizes the signaling molecule that's on the outside of the cell, and then the inside of the cell, the inside part of the receptor is actually an enzyme. Okay, so you have one part on the outside that recognizes the signaling molecule, and then the other part of the receptor is on the inside of the cell, and it's actually an enzyme. And many receptors are protein kinases. Okay, and if you remember back to enzyme energy and metabolism, kinases are enzymes that add phosphate groups to molecules. So in the case of these enzyme-linked receptors, that's what they're trying to show you here. So you have the receptor, the signaling molecule binds to the receptor, that activates this, which is a protein kinase. So this portion of the receptor is going to take ATP, take a phosphate group from ATP, and then add it to this protein, and then that activates this protein. Okay, so this was how that type of, that's what would happen in terms of the signal causing the receptor to be activated, and then the receptor activation leading to the first part of the signal transduction pathway. So this would be the first protein that the receptor communicates with on the inside of the cell. Okay, so again, not all but most types of enzyme-linked receptors are going to function as protein kinases, where they're adding phosphate groups to specific molecules on the inside of the cell. Okay, the second class of cell surface receptors that we're going to talk about are called G-protein coupled receptors. And that's what's being displayed on this slide. Okay, and the reason that these are called G-protein coupled receptors is that on the inside of the cell, these receptors are going to interact with a class of proteins called G proteins. So that's why they're called G-protein coupled receptors, because on the inside of the cell, they're going to interact with a G protein. And in this picture, I think it's purple and blue. This whole thing right here is the G protein. So this is the receptor, and then this is going to be the G protein that's coupled to that receptor. And the reason that they're called G proteins is because they bind a molecule called GTP, which is a nucleotide like ATP, but instead of having adenine as its nitrogenous base, it has guanine as its nitrogenous base. So a G protein can bind GTP, and then also the hydrolyzed form of that, which is GDP.
You said they bind to
GTP is a protein then?
No, G protein is, G protein is, did you say G protein?
GTP, what is GTP?
It's a nucleotide. So we said, remember how we talked about ATP? So GTP is the same thing except it has guanine instead of adenine. Okay, so what happens in terms of these receptors is the ligand will bind to the receptor, and then that binding causes the G protein to interact with the receptor. And once the receptor binds to the G protein is going to release GDP, and in its place, it's going to bind GTP. Okay, so initially, before the receptor is activated, the G protein is sitting here inactive and bound to GDP. The ligand binds. The G protein now interacts with the receptor. It causes the G protein to release the GDP and bind GTP. Okay, now once GTP is bound, that causes a change in shape that causes the G protein to break apart. And it has several different subunits. One is called the alpha subunit, and the other is called the beta gamma dimer. And so what happens is once it binds GTP, the alpha subunit disassociates from the beta gamma dimer. And then that's going to lead to the next part of the signaling pathway.
GTP an energy source as well?
GTP can be an energy source. In this case, it's not an energy source. In this case, it's acting as a signaling molecule. So in the case of this, when the signaling pathway is off, the G protein has GDP bound to it. And when the pathway is on, it has GTP bound to it. But it is used as a energy source inside cells too. Okay, so then what happens in terms of this signaling is once the ligand unbinds from the receptor, what happens is the G protein is going to hydrolyze that GTP, and that's what causes it to now be GDP, and then the subunits come back together, and then it's inactive again. So after you've transduced the signal, this part of the G protein, which is the alpha subunit, is going to hydrolyze that GTP. Now it has GDP bound to it, and then it reassociates with the beta gamma dimer.
So what's usually accomplished when this happens?
So we're going to go through a specific example looking at specific signaling pathways. So we're going to see how once you get these G proteins disassociating from each other, once the G protein disassociates into these subunits, we're going to see what they do. So we'll talk about a couple specific signaling pathways. Okay. So then the third category of cell surface receptors are ligand-gated ion channels. So these are the same thing as ligand-gated channels. But this is the third type of, just thank you, receptor. I don't know what my problem is today. What's that?
It's cool, it's late. Yeah.
So anyways, a ligand-gated ion channel is a receptor that is also a channel. So in this case, you already know what's going to happen with this one. The ligand binds to the channel opens up, and then the ions flowing into the cell are going to lead to the activation of a signaling pathway. Okay, so a ligand-gated ion channel is simply a receptor that responds to a ligand binding to it by opening up a channel in the membrane. Okay. So just really briefly, I did mention to you that, so those are the three different categories of cell surface receptors. I did mention that some receptors are located on the inside of the cell. And just one example that I gave you earlier was the receptors for steroid hormones. One steroid that's found that's used in animals is estrogen. And so the receptor for estrogen, as you can see, is actually present in the nucleus. And so for this type of signaling, estrogen has to enter the cell and then bind to the receptor that's on the inside of the cell. What's special about this is that whatever the signaling molecule is, so estrogen in this case, it has to be able to get across the plasma membrane. And the reason that estrogen can do that is because it's a steroid, which is a lipid, and so it can pass through the membrane without any aid. Whereas the other molecules that we talked about with the cell surface receptors, the ligands for those receptors are usually water soluble and are unable to pass through the membrane, which is why the receptor needs to be on the surface, because it can't pass through to signal on the inside. So it needs a receptor that'll recognize it on the outside and then become activated in response to that. Okay. And then this is just reiterating something we've already talked about. So what we're going to get into now is, or what we just talked about was what happens when a receptor becomes activated. So in some cases, the receptor is actually an enzyme, so when it becomes activated, the enzyme becomes active. In some cases, the receptor is coupled to G proteins, and what happens when the receptor becomes activated, the G protein disassembles into different components, and then that's going to go on to activate downstream signaling pathways. And then in the third case, what happens is the signal binds to the receptor, and then the receptor opens up as a channel, ions enter the cell, and then that leads to the signaling pathway. So then once you get the activation of the receptor, that's going to lead to a signal transduction pathway that relays the signal from the receptor to whatever the effector protein is. In this specific example, this is going to be a transcription factor that causes the production of a protein. Okay, and so you would have a set of molecules that relays the signal from the receptor to the effector protein, and then once you get to the effector protein, it causes a change in protein expression, and then that carries out the cellular response. Okay. No, in terms of that, that just goes back to something I've already told you. So you should know in general the components of a signaling pathway. And the three components are a receptor, a signal transduction pathway, and an effector protein. And then what they each do. So the receptor recognizes the signal. It's why the cell can respond to a particular signal. The signal transduction pathway relays the signal from the receptor to the effector protein, and then the effector protein carries out the cellular response. Okay. So in terms of signaling pathways, two things that we'll see that happen very commonly in terms of a protein being off and then being turned on during a signal transduction pathway. One thing that commonly happens is that you're going to get signaling by phosphorylation, and that's on the left here. Phosphorylation simply means adding a phosphate group. Okay, so you have this protein that's a part of a signaling pathway, it's off, then a kinase adds the phosphate group, or you would say the kinase phosphorylates the protein, and now the protein is active, and then you can have a protein phosphatase remove or dephosphorylate the protein and turn it back off. So what would happen in this kind of signal transduction pathway is once the receptor becomes activated, it signals to the first component of the signal transduction pathway, which is a kinase that then phosphorylates and activates another kinase and so on. Another common way of relaying signals is through the binding of GTP versus GDP. And this is the case for G proteins. So when the G protein has GTP bound to it, then the protein is on, but then when it has GDP bound to it, then, oops, I did it the wrong direction. So it has GDP and it's inactive. Then it releases GDP, binds GTP, now it's active, and then once it hydrolyzes that GTP, then it'll become inactive. And so what can happen in the case of signal transduction pathways is you have the G protein sitting there with GDP bound to it, and then the activation of the receptor causes the G protein to switch GDP for GTP, and now it's active, and then once the G protein hydrolyzes that, it'll become inactive again. But when it's active, then that's going to relay the signal down the signal transduction pathway. And then you don't need to know this slide, but it's just showing you gaps and GEFs are involved in activating and inactivating G proteins. Okay. But don't worry about this slide. Two other important things to realize about signaling pathways is that you have positive feedback that can happen in a signaling pathway, and you can also have negative feedback happening in a signaling pathway. And I think it's pretty self-explanatory. So what you have in positive feedback is A activates B, and then once B active, it can relay the signal farther down the pathway, but it also goes back and activates A. So that would be positive feedback. Okay, negative feedback is the opposite. A activates B, and then B ends up going back and inhibiting A. It doesn't happen right away. It happens after a certain amount of time. So that's positive feedback versus negative feedback.
So B would essentially go back and reactivate A.
In positive feedback, yes.
So you can do the process then?
Yeah. So the positive feedback is useful. Is a useful way of if you need something to happen rapidly. Positive feedback is one way to cause that to happen, because once you activate it, then it's this positive feedback loop that keeps it activated, and that'll cause something rapid to happen. Negative feedback is useful because you don't necessarily want the pathway on the whole time, so once you turn it on, you do want it to shut off at some point, and negative feedback would be one way of, once you activate it and you get the response you need, the negative feedback shuts the pathway down. Okay, so now what we're going to look at is, you asked the question about what happens once the receptor gets activated on the inside. You were specifically asking it for G proteins, but we're going to go through one case where a protein kinase cascade gets activated, and then we'll talk about a specific example where you get the G protein activated and what that can lead to. So the first example, so we're just going to look at specific signaling pathways. And the first signaling pathway that we're going to talk about, the signaling molecule is EGF. And EGF stands for epidermal growth factor. And one thing that it causes on skin cells, is that it causes skin cells to divide. So EGF is going to bind to the receptor. The cellular response is that the cell divides and gives rise to two cells. And in terms of this signaling pathway, it's going to involve what's called, the receptor is called a receptor tyrosine kinase. And so this is an example of an enzyme-linked receptor. So EGF is the ligand, and then the receptor is specifically called a receptor tyrosine kinase. It would simply be called the EGF receptor, EGFR. don't know if they have it that way. No, they just call it EGF receptor subunits. Okay, and in terms of this, this particular signal transduction pathway, and this is also for there's a lot of other signal transduction pathways that happen in the same way, but what you have is you have the receptor being activated, and then you have a set of relay proteins, and the relay proteins are going to connect the receptor to what's called a protein kinase cascade. Okay, so you have the receptor, relay proteins, and then a protein kinase cascade. And again, the relay proteins relay the signal from the receptor to the protein kinase cascade, and then you have the actual protein kinase cascade. And what happens in a protein kinase cascade is the different components simply phosphorylate each other. So this is it's hard to see it in here, but RAF phosphorylates MEK. That activates MEK, which then phosphorylates ERK. ERK becomes active and phosphorylates something else. So that's the protein kinase cascade. So it's a cascade of phosphorylations. And then the third part, so you have the relay proteins, the protein kinase cascade, and then you have the cellular response, which in the case of this signaling pathway is carried out by transcription factors that are going to alter the expression of genes. Okay, so what happens in the case of this signaling pathway is, I want you to remember the general, what I just went through, not necessarily the specifics, meaning I want you to know that in this case you have a receptor being activated, and then you have relay proteins, and then you have a protein kinase cascade. You should know what happens in a protein kinase cascade, and then that leads to the activation of your proteins that carry out the cellular response. Okay, so, but just to be a little bit more specific, what happens when EGF binds to its receptor? It actually causes the receptor to, so you have actually two subunits, and they kind of come together, and then they phosphorylate each other. So the receptor actually just adds phosphate groups to itself, and what happens is once it phosphorylates, there's a protein here that recognizes that phosphate group when it's added on. So it binds to the receptor when that phosphate group is added, and then that is bound to this protein, which binds to this protein, which activates it, and then this protein activates the protein kinase cascade. So you get GRB, SOS, and RAS are your relay proteins. And then your protein kinase cascade consists of RAF, MEK, and ERK. So then once RAS becomes active, and RAS is an example of a G protein, so when RAS is active, it binds GTP, and then RAS activates RAF, which is the first component of your protein kinase cascade. RAF adds a phosphate group to MEK. That activates MEK. MEK activates ERK, so MEK adds a phosphate group to ERK. That activates ERK. ERK then translocates into the nucleus where it phosphorylates and activates those two transcription factors, and then they change gene expression. And then in a skin cell, that's going to cause the cell to divide. And because the genes that are controlled by these two transcription factors affect cell division. So you would lead to the expression of proteins that are involved in cell division, and then the cell divides in response to that signal. Okay, do you guys have any questions about this general type of signal transduction pathway?
So like when mature cells grow, like that usually has it like some abnormality or something?
No. There are times when your body is going to want to have cells divide and replace other cells.
Well, like growth instead of division.
Oh, sorry, growth. So what was your question again?
So like when cells grow instead of like divide,
Mhm.
they're
That's also a signaling.
sort of abnormality.
No. You can have, so some, I can't remember which specific, so there's two ways of enlarging a tissue. So one would be making more cells, and other would be making cells grow larger. And there's some cases where you make the cells grow larger, and that's not abnormal. This is something that they would want. And also, you typically you get cell growth with cell division, because what you want is to produce, typically you want to produce two cells that are the same size as the parent cell. So in order to do that, before you divide, the one cell enlarges, then divides into two cells. Okay, do you guys have other questions relating to this pathway? Okay, so what we're going to switch to now is talking about what happens in terms of G protein coupled receptors. And one of the things that can happen with G protein coupled receptors is that you get the production of what are called second messengers. Okay, so so one thing that can happen in terms of what happens once a G protein becomes active is you get the production of what's called a second messenger. So what we're going to do is talk about second messengers first, why they're important, and then we'll talk about specific pathways. So in terms of messengers, the first messenger is the ligand. So that's considered to be the first messenger. And then these are things that can be produced in response to that first message. And so that's why they're called second messengers. And it gives you examples of second messengers. So cyclic AMP is a second messenger. And that is a nucleotide that we discussed earlier in the class. A molecule called diacylglycerol. Also one called inositol triphosphate. And then also calcium ions are all examples of second messengers inside cells. And as it says at the bottom of the slide, the advantages of second messengers is that they will amplify a signal, and also they will speed up the cellular response. And the speeding up part comes from the fact that most second messengers are small, which means that they can diffuse rapidly. So remember, the smaller a molecule is, the faster it diffuses. So second messengers move relatively fast through the cytosol. And then also amplification. So I'm going to come back to this slide in a second. But what happens is once the receptor gets activated, that leads to the production of not just one second messenger, but multiple molecules of the second messenger. So from one receptor, you get multiple molecules of cyclic AMP made, and then each of those molecules can go on to activate the next portion of the signaling cascade. And so in that way, it amplifies the signal that's occurring from the receptor. So speed and amplification of the cell. And then just in terms of specifically talking about what can happen in terms of cyclic AMP, that goes back to this slide, and how this can be associated with G protein coupled receptors is that there are different types of G protein alpha subunits. So remember we said that when a G protein coupled receptor becomes active, the G protein interacts with the receptor, but then the alpha subunit dissociates from the beta gamma subunit. And what happens in terms of some alpha subunits of G protein coupled receptors is they bind to this enzyme called adenylyl cyclase. And that's the enzyme that takes ATP and makes cyclic AMP. So what would happen in this case is you get the activation of the G protein coupled receptor, and then the alpha subunit of the G protein activates adenyl cyclase, then you produce cyclic AMP, and then cyclic AMP goes and binds to something, and then the signal gets relayed farther down inside the cell. Okay, do you guys have any questions about second messengers, why they're useful, or that one specific example that we've gone through so far about how a G protein coupled receptor can lead to a cellular response? Okay, then let's talk about this specific example. So this is going to be a signal transduction pathway that involves cyclic AMP. And you may have heard of the ligand in terms of this pathway. It's epinephrine. Have you guys heard of epinephrine before? Yeah, you have. It's both actually a hormone and also a neurotransmitter in your body, depending on who releases it. So if an endocrine cell releases it into blood, it's a hormone. And then if a neuron releases it at a synapse, then it's a neurotransmitter. So what happens in this case is epinephrine is the ligand, and then the receptor that it's going to activate in this case is a G protein coupled receptor. So what happens in this case is epinephrine is going to bind to the receptor. That activates the receptor, and what's not shown is originally the alpha subunit and the beta gamma dimer were together, but when the receptor becomes activated, they dissociate with from each other. Then the activated alpha subunit binds to adenyl cyclase, and that stimulates adenyl cyclase to synthesize cyclic AMP. And then what happens in this pathway is cyclic AMP binds to this enzyme called pKa, which is simply protein kinase A. And what you can see here, and this is going to be an example of allosteric regulation, cyclic AMP binds to these regulatory subunits, and then that releases the catalytic subunits of pKa. So cyclic AMP binds, activates this kinase. This kinase then phosphorylates the response protein. So whatever protein carries out the response is phosphorylated and its activity is changed. So that's just one example of what can happen once a G protein coupled receptor is activated, and then how that leads to a cellular response. Do you guys have any questions about that? Okay. And then this is actually taking it a step further and showing you the specific response that a skeletal muscle cell has to epinephrine. So it's going to lead to two different things in this case. It's going to lead to glycogen breakdown, and then also you're going to inhibit the synthesis of glycogen. So in this case, what's going to happen is epinephrine is going to result in an increase in the production of glucose in the cell, and then that glucose can be used as energy. So remember we said glycogen stores energy, but you have to break it down into glucose in order to use it. So this pathway is going to lead to, one, stimulating the actual breakdown of glycogen, but then also inhibiting its synthesis. And by doing that, you're going to raise the glucose concentration in the cell and the amount of energy. So what they're showing you here is that epinephrine binds to its receptor. That leads to the alpha subunit activating adenyl cyclase, which leads to cyclic AMP production. Cyclic AMP activates that enzyme protein kinase A, and then protein kinase does two things. It phosphorylates this kinase, which activates it, and this is going to lead to glycogen breakdown, the stimulation of glycogen breakdown. Phosphorylation of this enzyme inhibits it, and then that's going to inhibit glycogen synthesis. And then that's going to increase the glucose concentration in the cell. And you may have heard of epinephrine being the flight or fight response hormone. And so this would be a way of preparing a skeletal muscle cell for that flight or fight by providing it with energy in case it needs to become active really quick. Okay, do you guys have any questions about this specific example? Okay, and then we have one more example to go through. And this is actually going to involve several second messengers. So it's going to involve a second messenger called diacylglycerol, inositol triphosphate, and then also calcium. Okay, so in this pathway, what happens is the signaling molecule activates a G protein coupled receptor. That then leads to the alpha subunit of the G protein becoming active. And in this case, the alpha subunit of the G protein activates this enzyme called phospholipase C. And phospholipase C creates the second messenger called IP3, which is inositol triphosphate. Okay, so you get activation of the receptor, leads to the activation of the alpha subunit, G protein alpha subunit. That activates phospholipase C. That creates the second messenger IP3. IP3 then diffuses to the endoplasmic reticulum, and when it binds to the endoplasmic reticulum, it causes the endoplasmic reticulum to release that stored calcium. So remember we said that one thing that ER does is it stores calcium ions. Well, if IP3 is made and then binds to the ER, it causes that calcium to be released. And there's actually a bifurcation here that IP3 is also going to lead to activated PKC, which is protein kinase C. And then that's going to lead to part of the cellular response, but then also the release of this calcium, which is a second messenger, binds to proteins in the cell, and that activates the protein. So there are certain proteins that respond to calcium binding to them by becoming active. So in this case, it involves several second messengers. The first second messenger is IP3, and then I didn't mention it, sorry, the other one was diacylglycerol. That's the one that activates PKC, and then the third second messenger is calcium in that pathway. Okay, and then just as an example of this in smooth muscle cells, so if this was a smooth muscle cell, PKC is going to phosphorylate proteins that are involved in muscle contraction, and then that would lead to muscle contraction. Okay, do you guys have any questions about this specific signaling pathway? Okay, so in terms of the just announcements, and then we're done for the day. So you guys are probably aware of this. I know I am. Next week is spring break. But I kind of had to schedule the lecture exam for the week after spring break. But what I'm going to do is actually we're going to switch the lab and the lecture so that the exam happens on Wednesday. That way it's not right when you get back, and you have at least a few days, a couple of nights to, I wouldn't wait until this 2 days before the exam to study for it. But it gives you a couple more days to study for the exam after the break is over. So in terms of, so next week is spring break, but then the week after that you're going to have your second lecture exam, and that exam is going to be on Wednesday rather than Monday, and I'll of course send this out to you as a message later this week. And then in terms of this exam, what we're also going to have to do, because we're actually really behind in the course, is we're actually going to have to speed up. I know it may seem like we're going fast, but we're actually behind. So I'm going to have to speed up the rate at which I'm presenting to you guys so that we can catch up. So this exam is actually not going to include that much information because I'm actually a little bit farther behind in my other classes than this one. And so in terms of the exam that's going to happen, it's only going to cover the rest of cell structure and function that was not on that first exam. So the rest of cell structure that wasn't on that first exam, and I'll give you the specific slide numbers in the announcement, and then it'll include viral structure and reproduction, and then membrane function, structure, and synthesis. So only those three topics. The rest of cell structure and function, there's one, the energy, enzyme, and metabolism also. So sorry, 4. Rest of cell structure and function, viral structure and reproduction, membrane structure, synthesis and transport, and energy, enzymes, and metabolism, but not any of the, we did finish cell communication today, but I won't finish it for my other classes. I'll probably only get halfway through, so that will not be on the second exam that you guys take the week that you guys get back. And then of course you do have lab this week, and your first statistics are due in lab on Wednesday along with the pre-lab. So that's it for tonight. I'll see you guys on